الناس الاميننت جدا في كلينيكال كيميكال باثولوجي، وخصوصا كل الحاجات التست اللي طول النهار بنتعبه، التي ام اس والان جي اس وكل الحاجات اللي احنا بنحتاج لها جدا. فيعني very happy ومفكرك دايما عايزينك معانا في الجيناتكس دايما ان شاء الله.
ميرسي جدا دكتور. ميرسي جدا دكتور هالة، وميرسي دكتور ايمان وكل Okay, my name is Mohamed Abd Elmonem. I'm a professor of clinical pathology, Faculty of Medicine, Cairo University, and today we are going to talk about the extensive genetic sequencing for the early detection of genetic disorders in the neonatal ICU. What is genetic sequencing? If we can remember all, احنا عندنا النيوكليوتايد بيزز بتاعتنا في الدي ان اي بتاعنا اللي هي الادينين والثايمين والسيتوزين والجوانين. They form the basic structure of our DNA, and thousands of these bases together they can form genes. Each gene has multiple exons and multiple introns. The exons are the coding part that codes for proteins, and the introns are usually the silent parts. They do not code for proteins. Thousands of genes, they form a chromosome, and 23 pairs of chromosome, they form our DNA, our genome inside the nucleus of our cell. To give you an idea about the size spectrum of the tests that we do for genetic sequencing, we can look for certain small variant in a non-gene. If we know the causative or the pathogenic variant in a certain family, we can look for this variant specifically, and we usually do a small sequencing of one exon between 100 and 500 base pairs. We can also look for variants in a single non-gene. If the phenotype is clear enough, I can sequence a specific gene, and this usually between 1,000 and 50,000 base pairs of sequencing. We can look for variants in a panel of non-genes if the genes responsible for this phenotype are various, and this usually between 50,000 and 2 million base pairs. To increase the spectrum, we can look for variants in the clinical exome. The clinical exome are the genes that are linked to human disease, and they are about now 5,000, 6,000 genes. And the clinical exome means that we have to sequence about 5 million base pair of our genome. We can do a whole exome. A whole exome means we do all the exome, all the coding exons with the flanking regions between the exons and the introns, and we do sequencing of about 30 million base pairs. Or we can go for the big genome and look for the whole human genome. The human genome contains about 3 billion base pairs, which is a huge amount of data. Now we can talk about genetic variants, the types of genetic variants. Hello? Now we can talk about types of genetic variants that are normally in our genome. We have single nucleotide variants, which can be missense, just a single base change leading to or not leading to an amino acid change in the coding sequence. And sometimes it can lead to a nonsense variant. Nonsense variants means that stops the translation of the DNA sequencing into protein. There are indel variants which are either insertions or deletions. They are small insertions or deletions that can cause frame shift. The forming protein is not the same thing, just deleting one of two. Just deleting. Just deleting one or two, the in-frame insertions or deletions, just deleting one or two amino acids while keeping the sequence afterwards. We have also splicing variants. Splicing variants are the variants that occur at the exon-intron boundaries between the small region between the exon and the intron, and they may change the RNA transcription, leading to the formation of a defective protein. We have also copy number variants and big structural variants. These are large deletions or duplications that expand across multiple genes or sometimes across a large portion of a chromosome. And we have mitochondrial DNA variants in the mitochondrial genome. What is a pathogenic variant? We all see the reports that come for genetic diagnosis of patients, and pathogenic variant is defined, it's a probability. The pathogenicity, it's unless it's supported by functional evidence in a certain, in the literature, it's a predictive model. There is criteria or guidelines published from the American College of Medical Genetics, or the ACMG guidelines, that define the criteria needed for the different classifications I'm going to talk about now. Pathogenic variant, the variant is well established as disease causing in the database and literature. It's absent or very rare in population databases, and a wide consensus on the variant pathogenicity exists. In these cases, significant family segregation has been verified, and several publications support pathogenicity. For this type of variants, the probability of being pathogenic or disease-causing is more than 99%. Likely pathogenic variant, which a little bit different, the identified variants considered. Likely pathogenic variant, which is considered the probable cause of the patient disease, as it is a good fit for the patient phenotype. The evidence is strong. ميرسي، شكرا. شكرا. This is much better. Okay. Likely pathogenic variants, as we have just spoken, the identified variant is considered as the probable cause of the patient disease. It's a good fit for the patient phenotype, and the evidence is strong, but there is still a small degree of uncertainty. The variant is also absent or very rare in population databases, and its probability of being responsible for the disease is more than 90%. Variant of uncertain significance, and here we come to the conflicting type of variant. Is typically rare. It could be predicted to be deleterious, and the gene has an association with the phenotype. However, the evidence is not yet enough to swing it towards being pathogenic or being benign. The chance of being pathogenic between 50 and 90%. Likely benign, it's not likely to be the cause for the disease, and the chance of being pathogenic is less than 50%. Benign, the chance is less than 5%. As I just said, the criteria for judging the pathogenicity of variants is unlisted by the American College of Medical Genetics, and we almost all of us follow these rules. What I just want to clarify that the variance of uncertain significance is not one type of variance, it's a spectrum. Some of these variants are very close to being pathogenic, and some of these variants are very close to being benign. So we have to be very cautious when reporting variants of unknown significance. Now we are going to talk about extensive genetic sequencing. What is meant by extensive genetic sequencing? Extensive genetic sequence simply means sequencing done by the next generation sequencing, generating a huge amount of data that needs interpretation at the bioinformatic level and then at the clinical genetic level. There are main genetic panels, genetic panels, and we order usually genetic panels when we suspect a phenotype that has been reported with a multitude of genes, a whole lot number of genes that's very difficult to do by Sanger sequencing. When screening of most common causing genes by Sanger sequencing or DNA hybridization has revealed no results, and when there is already an available gene panel that satisfied my need to cover the patient phenotype. Then whole exome, when the phenotype is so complex, I don't have a clear indication as to which way I should go. When the phenotype corresponds to a panel of genes that are not available to me or in my country, and when the panel of genes I could find is not inclusive of recent genetic additions to this phenotype. And more recently, the concept of actual genetic panel has been more or less abolished and replaced now by the concept of virtual genetic panels. The technique I do is a whole exome sequencing, and then the analysis I do is for specific genes that satisfy the patient phenotype. And whole genome, when we do whole genome, when you have a very strong suspicion of a genetic syndrome and the whole exome came back negative, or when you suspect the etiology behind your patient syndrome are big structural variants, because whole exomes are defective in this regard. Now we are going to talk about some studies that performed extensive genetic sequencing, either whole exome or whole genome, in the neonatal ICU. For example, Meng et al. 2017 conducted clinical exome sequencing on 278 unrelated infants within the first 100 days of life, admitted to Texas Children's Hospital in USA. Genetic diagnosis was achieved in 102 infants by exome sequencing with a diagnostic yield of about 37%. The diagnosis affected medical management in 53 of the 102, which is 50% of diagnosed infants and 19% of total. Three-year exome sequencing in their study revealed a molecular diagnosis in about 50% or 51%, which is a little bit higher than the single individual approach. In their study, the diagnostic yield ranged between 20% in abnormality of the endocrine system to 65% in abnormality of blood and blood-forming tissues, but averaging around 40%. Another study, Suzuki et al., conducted a prospective study in 17 tertiary care centers in Japan, conducted between 2019 and 2021. Critically ill neonates and infants less than 6 months of age were recruited undergoing either conventional whole exome, whole genome, or both. When the whole exome came back negative, they ordered whole exome and see what comes up. Of the 85 patients screened, 41 had positive results, about 48% diagnostic yield. And based on the primary symptoms, patients with metabolic phenotypes have the highest diagnostic yield, about 67%, followed by renal 60% and neurological phenotypes. Among patients with a confirmed genetic diagnosis, about 50% had significant changes in clinical management plans, and 23% of the total. Kingsmore et al. 2019 screened 213 ill inpatient infants less than 4 months age at the Raleigh Children's Institute for Genomic Medicine at San Diego, California, USA, and compared the performance of rapid whole exome to rapid whole genome sequencing using a pipeline of rapid sequencing and AI-assisted interpretation of next-generation sequencing data at their institute, the Raleigh Children's Institute, that gave back results in an average of 5 to 14 days. The normal span for receiving diagnostic outcome for whole exome and whole genome sequencing following the normal pipeline is about 6 to 8 weeks, more or less, give or take. But they proposed a pipeline that's dependent on a lot of sequencing machines. In the lab, they have like 50 next generation sequencing, and they have huge amount of personnel working on this project to produce the rapid diagnostic outcome. The diagnostic performance of the rapid whole genome sequencing and rapid whole exome in their study were similar, more or less, giving a diagnostic yield of 19-20% respectively, as was the time to result, with a median of 11 and 11.2 days. Sorry. In their analysis, the three samples didn't significantly add to the diagnostic yield, but shortened the time needed for the confirmation in positive cases. Because when I get a positive result with a novel variant, a variant that hasn't been reported before, I need to confirm this variant in the parents of the proband. Thus, a trio analysis from the start is very much time saving. Another study by Demak et al. 2021 conducted rapid whole genome sequencing in 184 infants, but less than 1 year, at 5 different hospitals in California, USA, with the same hospital responsible for the previous study, Rady Children Institute, as the project coordinator. Of the 184 infants, about 40% received a diagnosis that explained their admission in a median time of 3 days. They accelerated the time frame of their analysis, which is, again, it's a lot of effort to do that. And in 58 individuals, 32%, rapid whole genome led to changes in medical care approach and management. This is just to give a hint about the inclusion and exclusion criteria in similar types of study. The inclusion criteria are very simple. It's an acutely ill neonate admitted within the time frame of their study. They clarify that it should be within 1 week of hospitalization or within 1 week of development of abnormal response to standard therapy. But the exclusion criteria is the more informative. Perhaps Dr. Noor in the next speak will talk more about the clinical criteria to include and exclude neonates for extensive genetic sequencing. But they have to exclude all the common causes that's not genetic. In another study, Moriello et al. Conducted a meta-analysis from 18 retrospective and prospective study of both whole exome and whole genome sequencing in pediatric intensive care units, which has demonstrated an average diagnostic yield of about 40% and an impact on clinical management in more than 20% of patients tested. The highest clinical utility was in the neonatal setting, with a diagnostic yield of around 50% on average, when cases are properly selected, of course. They concluded that extensive genetic sequencing, when indicated, should be the standard of care for patients in the NICU. Now let's talk about our status in Egypt from these techniques. Genetic next generation sequencing technologies are getting a foothold in Egypt now. It's not as fast as we hope, but we are slowly advancing. The causes for this lag or for this slow advancement are known to us all. Number 1, 2, 3, and 4 are funding. It's very, very expensive to do that. And there are also other logistical issues, including the supply chain, the maintenance from the supply companies, and the technical support. It's very weak still in Egypt. The good news is that a considerable part of the Egyptian Genome Project launched last year will be dedicated to clinically based cases, particularly in the pediatric age. Through the collaboration between the Egypt